All right, today we're cracking open a really foundational piece of neuroscience. It's a 1993 paper from Roland Strauss and Martin Heisenberg that dug into one of the brain's most basic jobs, telling the body when, why, and how to walk. It's a real masterclass in scientific detective work, and it helps shape how we think about the chain of command between the brain and the body. So, let's get into it.
So let's start with something kind of weird, but it's a super important biological fact. You've heard the phrase, running around like a chicken with its head cut off, right? Well, it's not just a saying. A chicken, or a cat, or even a decapitated insect can still make basic walking movements. And that's because the nerve circuits in the spinal cord, or the insect equivalent, can run the basic walking software all on their own. They're called central pattern generators, and they can just produce these rhythms without any input from the brain. So if the body can handle the rhythm of walking by itself, what is the brain even for? What's its job? And that question gets us to the absolute core of this whole paper. On one side, you've got these lower motor programs, the automatic stuff, the rhythms from the spinal cord. But on the other side, you have higher control. Now this is the brain's turf. The authors put it perfectly. They said, what makes behavior so interesting is not so much what the behaving organism actually does, but why, when, and how it does it. That whole why, when, and how, that is higher control. It's the decision to start walking towards something, the choice to speed up, and that internal drive to just keep going. This paper is basically a manhunt for the physical location of that higher control. To track down this control center, Strauss and Heisenberg laid out this brilliant 5-part investigation. First, they identified their prime suspect inside the brain, the ghost in the machine. Then they brought in their key witnesses, a whole lineup of mutant flies with really specific problems. After that, they put these flies through a series of behavioral challenges, and that led them to the ultimate mind-body test to really isolate the problem. And finally, they could deliver their verdict on what they found, the brain's walking CEO. Okay, part 1, the brain's ghost in the machine. Our whole investigation is going to take place inside the brain of the humble fruit fly, Drosophila melanogaster. For over a century, the fruit fly has been this absolute powerhouse for genetics research. Why? Because it's simple enough for us to actually study, but it's still complex enough to tell us some really fundamental things about biology, including how a brain actually creates behavior. So the researchers' attention really zeroed in on one particular structure, the central complex, or CC for short. It's tucked right in the middle of the fly's brain, and it was a suspect because of its really unique anatomy. It's this collection of modules with this strikingly regular, almost crystal-like structure. And in biology, when you see that kind of precise, repeating architecture, it's often a big hint that it's involved in some kind of complex computation, maybe like coordinating movement or navigating through space. It was just the perfect candidate for a higher control center. On to part 2, a lineup of mutant flies. So how in the world do you prove what the central complex actually does? You got to remember, this is 1993. We didn't have fancy tools like optogenetics to just turn neurons on and off with a flash of light. So the researchers turned to this classic, incredibly powerful tool in fruit fly genetics. They found flies that were already broken in exactly the way they needed them to be. They used a method called genetic dissection. The logic behind it is just beautifully simple. If you want to know what one part of a car engine does, what do? You carefully take it out and see what stops working. It's the same idea here. By finding a bunch of flies that were born with genetic mutations that specifically damaged the central complex, the scientists could just watch their behavior and figure out what the CC is normally responsible for. It's like using a genetic scalpel to reverse engineer the function of the brain, one broken piece at a time. Now, before we look at all the broken ones, we need to know what a good one looks like, right? This is our baseline. It's a diagram of healthy, or wild-type, central complex. And you can see its intricate, really symmetrical structure. You've got the bar-shaped protocerebral bridge up top, the big fan-shaped body below it, and that smaller ellipsoid body tucked inside. This is the blueprint, the fully operational version that our researchers started with. Okay, now meet the first member of our mutant lineup. This is the no-bridge mutant, and what's just incredible here is how precise the defect is. If you look at that protocerebral bridge at the top, there is a clean gap right down the middle. It's not just a mess, it's a specific, targeted break. This is that genetic scalpel in action. It gave the researchers a way to ask, okay, what happens when just this one piece is messed out? And just for contrast, here's another key suspect, the central body defect mutant. The damage here is, as the paper says, profound. The lower parts of the CC, the fan-shaped and ellipsoid bodies, they're all fused together into this disorganized, fragmented mess. You know, having this whole range of mutants, from the subtle break and no bridge to the total disarray here, that was a key part of the strategy. It let them ask a really critical question. Does the severity of the brain damage actually line up with how severe the behavioral problems are?
And that brings us to part 3, putting flies to the test. So with their lineup of normal flies and mutant flies all assembled, it was time for the fun part. Time to see what they could and couldn't do. The researchers designed this whole battery of clever behavioral experiments, these gauntlets, to systematically measure different parts of walking and figure out exactly what was going wrong in the mutants. The star of the show was this really elegant experiment called Briden's paradigm. So picture this. A single fly is placed on a tiny round platform. It's surrounded by water, so it can't just fly or walk away. And in this totally stark environment, the only things it can see are 2 big black stripes on opposite sides of the arena. Now this setup is brilliant because it's testing more than just walking. It tests motivation, it tests goal-directedness. A healthy fly won't just wander around randomly. It'll spontaneously and persistently walk back and forth between those 2 visual landmarks. And the results, I mean, look at this. They are just stunningly clear. On the left, you see the path of a normal wild-type fly. It's purposeful. It's a clean shuttle run, back and forth between the stripes. Then on the right, that's the path of a fly with a defective central complex. The path is just erratic. The fly wanders aimlessly, it stops for long periods of time, and a lot of the time it doesn't even make a single crossing. So the basic ability to move its legs is still there, but that higher level purpose, the goal-directedness, the motivation, it's just profoundly broken.
And you can see that crucial point quantified in this data. This chart compares walking activity at the very beginning of the 15-minute experiment to the very end. The wild-type fly starts strong and it ends strong, maintaining a really consistent level of activity. But now look at the CC mutant. It's a totally different story. It might start walking with some energy, but its activity just collapses over time. It basically gives up. The drive to keep walking just fizzles out. And this wasn't some slight difference. It was a huge decay in motivation.
Now, any good scientist is going to think about other explanations. You might be wondering, well, couldn't the mutant flies just be getting tired faster than the normal ones? The researchers thought of that, and they came up with the perfect control experiment. They took a mutant fly that had stopped walking, let it rest for a minute, and then just picked it up and placed it right back in the center of the platform. And the result? The fly's walking activity immediately shot back up to its initial high level before decaying all over again. And that proved it. This wasn't a problem of muscle fatigue. It was a failure of the brain's command system to keep the behavior going.
All right, let's move to the fourth and, honestly, the most conclusive part of this whole investigation, the ultimate mind-body test. So all the evidence so far is pointing straight at the central complex. But there's this critical, nagging question in genetics. Sometimes a single gene can have effects on lots of different parts of the body. So how could they be 100% sure the mutation wasn't causing some subtle, invisible problem in the fly's legs or muscles? This is the final hurdle. To prove their case beyond any shadow of a doubt, they needed to find a way to surgically separate the effect of the mutation in the brain from any potential effect in the body. They needed to do, essentially, a genetic mind-body swap. And incredibly, they had a tool to do just that. They used this breathtakingly clever technique to create what are called gynandromorphs, or mosaic flies. Now, through a whole series of genetic tricks, they managed to engineer individual flies whose bodies were a patchwork of 2 different kinds of cells. Some cells were genetically mutant, and other cells were genetically normal. And this let them create the 2 dream subjects for their experiment: flies with a mutant central complex in an otherwise healthy normal body, and the reverse, flies with a healthy central complex in a genetically mutant body. This is the definitive test. And here it is, the smoking gun. The first result from these mosaic flies, showing walking activity over time. On the left, a fly with a mutant brain but a perfectly healthy wild-type body. And look, it shows the exact same decay and motivation that they saw in the full mutants. Now, on the right, a fly with a healthy wild-type brain but a body made of genetically mutant tissue. And what does it do? It walks with sustained, high activity, just like a normal fly. The behavior followed the brain, not the body. Case closed. And here's the final nail in the coffin. They also looked at walking speed, or what they called walking vigor. The result was identical. A mutant brain, even in a perfectly good wild-type body, produced slow, hesitant walking. A wild-type brain, even in a mutant body, produced fast, vigorous walking. The conclusion was now totally inescapable. The problems with walking activity and vigor were caused specifically by the defects in the central complex and nowhere else. So, this brings us to our final section, the brain's walking CEO. After this incredibly thorough, just elegant investigation, which systematically ruled out every other possibility, Strauss and Heisenberg were finally ready to deliver a clear verdict on what the central complex actually does. And the verdict was crystal clear. The central complex acts as a higher control center for locomotion. It's kind of like the CEO of walking. It doesn't work on the factory floor, physically making the step-by-step rhythm happen. That's handled by the lower nerve centers. No, the CC sits up in the executive suite and makes the big strategic decisions. It controls walking activity, that's the decision to start and the motivation to keep going, which is what we saw just disappear in the mutants. It controls walking vigor, the speed and the power of each stride, which was way lower in the mutants. And it controls complex maneuvers, the starts, the stops, the turns that let an animal actually navigate its world with purpose.
I think the authors themselves summed up their landmark finding with this beautiful scientific precision in their conclusion. They wrote, "The pattern generator for undisturbed straight walking is located in the ventral ganglion. The readiness to use it, the vigor of the strides, are contributed by the CC in the brain." It's just the perfect, concise statement of that critical division of labor between the body's machinery and the brain's command.
This paper just so elegantly proved that even a seemingly simple act like walking requires a dedicated manager in the brain to give it purpose and drive. And in the decades since, we've learned the central complex is even more sophisticated than they knew, acting as an internal compass and integrating all sorts of sensory information to guide navigation. This 1993 paper really laid the groundwork for all of that. And it leaves us with this really profound question. If a tiny fruit fly has a dedicated CEO just for walking, what other specialized, high-level executives are running the countless complex shows not just in their brains, but deep within our own?